She received her BS in computer science, graduating cum laude from Arizona State University before being accepted into KCU's Doctor of Osteopathy program. She's been an exemplary student at KCU, earning Honors Designation in internal medicine, pediatrics, surgery, and psychiatry on her COMAT exam, along with KCU's COMLEX I Excellent Scholarship for scoring in the 90th percentile on her exams. During the quarantine and the pandemic, she organized and led a student research project summarizing the COVID-19 pandemic, thus creating our comprehensive document we have on the Canvas site. And like I said, she's here today to give us a second Journal Club presentation. So welcome, Jess. I will turn it over to you.
All right, thank you, Shelley. I will go ahead and show my slides.
Looks good.
Sorry, I'm just looking for I forgot where I was, the show slides thing. Okay, sorry about that.
Top right corner, maybe, where it says present.
I think I have it. Do you guys see it?
That's perfect.
Okay.
Yeah, you're good.
All right, so this presentation is on COVID-19 and adaptive immunity. Particularly, I am presenting two articles which were investigating the role of T cells and their potential for cross-immunity between COVID-19 and seasonal coronaviruses. So as background, COVID-19 infection is a mild or even asymptomatic illness in most people who wind up being infected with the virus. Currently, the CDC's best estimates are that somewhere between 30 and 70% of people who get infected are actually asymptomatic. And a high proportion of the people who do have symptoms wind up with a mild upper and or lower respiratory illness with or without GI symptoms. However, a small percentage, which is about 5% of the cases that they identify, experience a severe pneumonia, requiring respiratory support, sometimes go into respiratory failure, needing ventilatory support and intubation, and particularly among those who need ventilatory support, some of them wind up dying, a fair percentage of them. And so there have been a lot of efforts in the past to identify, which risk factors, like what they are for developing severe disease. But even when they have done this, that has not fully explained the wide heterogeneity and these big differences in the presentation of the illness from person to person. So I just kind of created this slide as an example because particularly, I'm in my clinical years, and this is something that, you will see with this virus, is that there's just this really, really wide spectrum of disease severity. So I just kind of gave an example, there's a 33-year-old female and a 68-year-old female. Several of them have, both of them have a couple of the risk factors. The 33-year-old doesn't have too many besides being pregnant. But the 68-year-old, winds up being identified because she was screened because she had family members who were positive, and she never winds up having symptoms at all. Whereas the 33-year-old needs an emergent cesarean section and weeks of mechanical ventilation before she eventually begins to recover. And so a lot of effort has been made to determine what exactly is the role of the immune system in all of this, because the immune system, it's most important possibly the least understood still overall in terms of its role in containing the pathogen, because that is what the immune system is supposed to do, is prevent the virus from, wreaking so much havoc on the body that you wind up, seriously ill or dying. So the immune system, we have innate immunity and we have adaptive immunity. The innate immune system functions to help prevent entry of the virus into cells and into tissues. And the adaptive immunity assumes a role when the innate immunity fails to do this. And so there's two major branches of the adaptive immunity, which is the B cells and the T cells, and both of which are capable of forming memory. And I'm just giving this quick review of the immune system because it helps the further articles along kind of make more sense and helps refresh people. The innate immune system, it prevents pathogens from entering into cells and tissues, is its main purpose. And it assists the adaptive immune system when a pathogen does manage to get past the original barriers. B cells are primarily responsible for antibody secretion, and antibodies also help prevent pathogens from entering cells, and they make the pathogens easier to destroy and activate other innate immune cells. And then with the T cells, there's several commonly discussed types of T cells, but in particular, the T helper cells activate other branches of the immune system, and the T killer cells are responsible for killing of infected cells. And so this just kind of goes over the little pathway that the activation of in which the activation of T cells occurs. So you have these little innate immune cells, which gather some of the pathogens and present them to the CD4 cells. The CD4 cells, which are capable of reacting to what's presented, wind up proliferating and becoming effector cells. The same thing happens with CD8 T cells, and then these cells, if they're CD8, they go and they find these antigens which are presented on infected cells and effectively kill them so that the virus can no longer use them. And the T cells, the CD4 T cells, help activate the rest of the immune system. And once again, both of these branches are capable of memory as well. And so we've known for some time that, different types of immune deficiencies tend to have different types of consequences in terms of the infections that people who, say, for example, are born with these deficiencies are prone to. So for example, with T cell deficiencies, T cells are very important for the clearance of viral infections. B cells are also important, but T cells, because viruses are intracellular, play a very important role against these types of pathogens and any sort of microbial pathogen which tends to take shelter inside of cells, so to speak. And so another thing to know about the T cell immunity is that it does change with age. The T cell arm matures primarily during the 3rd trimester of pregnancy. And so it is mostly mature. The thymus is mature at the time of birth. Technically, the T cells are not out in the periphery yet, but in terms of your T cell immunity's, say, adaptability, it's really at its peak around the time that you're born. And in children and in young people, the thymus plays a significant role in the generation of what we call the naive T cells, which are the T cells which are not already reactive per se to another pathogen. They're not a memory cell. They're just cells that are generated in order to be able to react to a pathogen when it comes. And so naive T cells are really at their peak when you're at your birth too. Memory T cells come after you've experienced infections. They can also come from allergies or any sort of immune reaction to a substance. And those increase throughout life. And naive T cells tend to decrease. The thymus in adulthood begins to play a much less significant role in the generation of these naive T cells. And around the late teen years and early adulthood is when the thymus begins to involute. And after this point, you have the body generates new T cells via peripheral generation. And this is important because it plays a role in like some of the theories about why the T cells and stuff begin to become less responsive with age, because it's believed that it is done via replication of existing naive T cells is how the new ones are generated. And then around the age of 65, both naive T cells generation and responsiveness begins to significantly decline. So there was a lot of like initial research and focus in the scientific community and the media on the role of antibodies in protection against SARS-CoV-2. They did find that they have found that nearly all people who are infected will have an antibody response at some point. The authors in these studies that I'm discussing were saying that antibodies to previous coronaviruses do not seem to play a significant role in protecting against COVID-19. However, I wasn't actually able to find the research that they were citing there, so I would just kind of take that with a grain of salt, that statement. And then in terms of, a lot of concern has been over, whether this antibody-mediated immunity, which does occur, lasts. And that's still pretty up in the air. But there have been some early studies which have found a waning and falling off of these antibodies that are generated as early as the convalescent phase, particularly in people who had a really mild or asymptomatic infection. All right, so this first article was an investigation of the T cell immunity in people who have had a prior COVID-19 infection, a prior SARS infection, and people who have never been infected with either. So this study was generated and it looked at the reactivity of T cells as opposed to antibodies to SARS-CoV-2 in donors from 3 categories. One of them was confirmed prior SARS-CoV-2 infection. The other was confirmed prior SARS infection and unexposed individuals. And they specifically tested for T cell reactivity to the nucleocapsid protein and to non-structural proteins, which was the NSP7 and the NSP13. And I believe these have been demonstrated in prior studies to be highly reactive, or antigens which the body tends to produce antibodies and T cell reactivity to. So they first looked at the responses of people who have recovered from COVID-19 infection, and they looked at the ability of they looked at the T cell responses to COVID-19. And what they found was that all of these individuals here had reactivity to the N1 and N2 protein. And some of them had the reactivity to non-structural proteins as well. Sorry, were there any questions? Never mind, that was just a mic open. They then also looked at the responses of patients who had recovered from SARS to the SARS-CoV-2 proteins. Whoops, sorry. And they found that there was some cross-reactivity between these memory T cells and to this N1 and N2 in people who had been infected with SARS but not infected with SARS-CoV-2. And so they also additionally found in their study that these were memory T cells, which had which were still, responding effectively 17 years later after a SARS-CoV-2, or sorry, a SARS infection. They then also compared the SARS-CoV-2 responses in people who had not ever been infected with SARS or SARS-CoV-2. And they took T cells from these donors and looked to see, what they were reactive, if they would react to any of these antigens. And what they found here, which was a significant finding, was that in unexposed individuals, approximately 30% of them did seem to have pre-existing T cells, which reacted with these well-studied N and NSP epitopes of SARS-CoV-2, which suggests that there may be some degree of, in this certain percentage of individuals, there may be some degree of cross-reactivity from prior infections of some sort. And so some of the conclusions that I had was that patients who were previously infected with COVID-19 were shown to have T-cell responses after recovery. They also had strong T-cell responses to the, people who had recovered from SARS infection had responses to the S-spike protein 17 years after infection. And they were finding that about 30% of normal individuals had robust memory T-cell responses to SARS-CoV-2 epitopes. And these T cells in these individuals seem to have, I forgot to say this earlier, a stronger predominance for the non-structural proteins overall. Some of the limitations of the study was that it was relatively low power because the number of participants studied, I believe, was 60 something. And of course, this doesn't determine whether these T cells are truly protective in any sort of way. So it really raises, in some ways, more questions than answers, because it raises questions like, can these T cells provide an actual immunity to infection or immunity to future infection, or perhaps, like for example, with the RSV virus, would it simply be a milder illness if somebody was reinfected, or would there be nothing at all? And it doesn't, if somebody were to be reinfected, would this person be contagious? It's just really a lot of different questions that it asks. It also does not identify the T cell subtype, which was reacting, which was definitely a limitation of the study, because we don't know if this was CD4 cells, CD8 cells, we just know that it was T cells. And that's something that the second study that I'm covering attempts to look at. So are there any questions so far?
Any questions for our speaker? Shelley Boyer.
Thank you, Jess. for that presentation. I just wanted to say I appreciate that you pointed out some of the issues, especially that they did not sort of determine the different types of T cells. So I think that would have made a difference maybe in some of the results had they been a little bit more specific in what it was they were looking at. But I appreciate that you made sure to point those things out. That shows that you definitely thought about the paper and ways that they could have made that better. So thanks for the presentation. I'm looking forward to the next one.
Okay.
Dr. Schneider?
Hi, Jessica. Very nice presentation. Jessica, in the literature, there is what you've shown that there is a very stable T cell response, right? So which is having a memory cells and which is protective immunity, technically speaking. But most of people who are tested on the immune response and memory using antibody test, right? So, and there is information in literature saying that people usually losing or having no memory cells, no memory B cells as they measure. So here, this is what important issue we come to. We have a antibody measurement as a surrogate marker of immunity, because cell-mediated immunity absolutely required for elimination of viral infections. Antibody supplement immune responses, right? So therefore, here, I'm glad you presented it, because I remember some of my colleagues even didn't understand what I was talking about. Our first meeting, when I just explained them why we don't have immunity against viral infection. We measure antibody response. We need to measure cell responses, T cell responses, which are not so sensitive. And formation of memory T cells are not sensitive to high level of IL-6, whereas B cells are very sensitive to that. So that is bringing the point that if we have, which we don't unfortunately have, reproducible or cheap method to test cell immunity, right? So that's what we need to go and measure. Antibodies, it is surrogate to the point that you prove with this data that most of the patients do have immunity, T cell mediated immunity. B cell is not reflecting exactly what is, because first of all, which antibodies they will measure, you clearly understand that if they would measure IgE antibody, that will reflect that there is T cell response. But from the beginning, people even don't worry about measuring what isotype they were measuring. They were measuring IgM antibodies. IgM antibodies could be produced without T cell response, as you know, because you had immunology. So in all of these things which people still kind of missing, I'm bringing it again. That is what we need to measure, but it takes 3-4 days before you kind of determine T cell response to any viral antigens. Unfortunately, it's not a massive screening technique. All right, so but that is the proof the point. So what I wanted to say based what you presented here. So be careful looking at claims about B cell response and antibodies and simply expropriate it to the general immunity against viral infection, which may not be. Because again, B cells are extremely sensitive to formation of memory. B cell is inhibited in the high levels of interleukin 6. And in opposite, it stimulates memory cells for CD4 cells. All right, so that is what your data is meaning for our interpretation of clinical data, where physicians just look, oh, antibodies. When you ask which antibodies, they say, oh, just antibodies. Yeah, so, but you know what we're talking about. It's right, so very nice that you presented this data. Thank you.
Okay, yes, and I find some of all this stuff about, examining the responses of different branches of the immune system to this virus and even to other viruses to be like very interesting overall, as a topic, because it's kind of like some viruses, when you get infected, you get infected one time and then your body mounts the immune response and then you never catch it again that we know of. Whereas there's other viruses like, one that I'm learning about in my NICU rotation is RSV, where people, become infected multiple times throughout their lives, even after mounting antibody and T cell responses against the virus. With the main caveat being that after the first infection, it tends to, in general, be a mild infection that essentially gives you a common cold. And so it's like the immunity that is developed, is like partially protective.
I see Dr. Johnson has his hand up. Mike?
Thank you, sir. Jessica, thank you very much for the presentation. I really enjoyed it. I have to excuse I need to let you know that I missed the last 3 minutes or so because I had to take a call, but perhaps this is covered. I don't know. My question to you is from an immunological perspective. You took us down took us down a road and explained a lot of those components to T and B cells and adaptive and innate immunity. My question to you is, from a clinician's perspective, do you feel that this immunological component is a contributor to the variations of presentations that these people are having, all the way from an asymptomatic positive test to minimal symptoms and serology, sometimes even deteriorating to worsening of symptoms and death? We're all over the board on these presentations of which patients are going to fly or walk. And my question to you is, in short, do you feel that the immunological data and information is partly accountable for the variations of presentations of this virus?
I mean, personally, I think that the evidence that has been coming out, particularly now that they're examining these T cells and the T cell immunity and potential for cross-reactivity, I think that it certainly would make sense that, especially like in an older population, having memory T cells which already react to the virus and help contain it and help appropriately activate the immune system before the virus actually causes significant disruption. I believe that definitely could wind up being, a factor in how sick people got. Another thing that, I kind of like covered at the beginning too was the number of naive T cells and naive T cell production that occurs in the body throughout lifetime, and how particularly right around the age of 65, on average, the responsiveness of T cells and other components of the immune system tends to start to decline. And so the immune system is less able to adapt to a new pathogen. But the number of memory T cells and memory B cells and plasma cells tends to increase throughout life. So if there were cells that could already interact with the virus due to, say, exposure to another coronavirus that you've had in the past, that could perhaps protect somebody who otherwise might become fairly ill.
Thank you.
But that's just speculation. from
I
me.
understand that, but that makes sense. That's there's a lot of variations. People are winding up, and it's hard to get a good handle, let alone wind up knowing how to treat them.
Dr. Schneider,
sure.
you have your hand up?
Sir. Well done. A, triple A, whatever. So, Michael, it's simply, we with age by 60, 70, and 80, which is the groups are most susceptible to this infection, and actually lethal outcome of this infection, because by that time we do not have cells which have a repertoire to recognize this new antigen. Our immunity completely by 70-year-old depends what this patient seen in the previous life, and they have only repertoire of memory cells. And that's why this is what the image what Jessica shown, where there is inverted relationship between number of naive cells and memory cells, indicates those repertoire is exhausted and you have whatever you have. And this group become vulnerable. And the more you proceed to 60, the less number of naive cells and chances to be recognized at right cells. Jessica, I just was, jumping on my chair, listening your answer. Thank you.
Thank you, Alex.
All
Let's
right,
go ahead
so
and move forward now.
Okay. All right, so the second article came out about a week ago. I think it's the 6th of August, and it kind of builds on the first study that I presented. And they had actually, done this research because of there were several studies which were similar to the first one that I presented, which were finding this, potential role for T cell, cross-reactivity. And so this study takes a further look into the potential for this. And it also looks at whether there could be some correlation between T cells, which are reactive to other coronaviruses, which circulate through humans through the seasons, and reactivity to the COVID-19 virus. All right, and so this study looked at kind of unexposed donors and SARS-CoV-2 recovered donors. And what they did was they took T cells and blood from the people who have never been exposed to COVID-19, which I believe they did by taking samples from people before the pandemic began. And they basically went and defined all of these epitopes of the SARS-CoV-2 virus, which the T cells from these unexposed donors could potentially bind to. They also went further on the previous study and investigated the subtype of the T cells which were responding. And they measured the strength of the response. So this study was really pretty in detail, and it was kind of hard. It was difficult to get a grasp on it first, from an outsider's perspective. But as I, had elaborated earlier, what they did was they found different epitope repertoires, which the T cells could bind to, and they defined them. And these were T cells from unexposed donors, and they found 64 to the genome spike, and to the spike protein, and 76 to other areas of the genome, which they called CD4S and CD4R. And then in these diagrams, they looked for homology of these sequences with other seasonal coronaviruses. And they also examined the degree of immunogenicity that was created. And then they used this to select some of their kind of epitomes of interest, so to speak. And so in here, they compared the CD4 plus C cells of unexposed donors that recognize these SARS-CoV-2 epitomes, and they also looked at the epitomes that were being bound by people who had had the virus previously, and looked to see if any of these unexposed individuals had T cells which could already respond and bind to the same epitopes that we were seeing with people who had a prior COVID-19 infection. They also elaborated on which type of immune cells were responding. For example, as you can see in B and C, the cell types that were responding in both the unexposed individuals and the people who had COVID-19 in the past were effector memory cells. So these were memory T cells that were responding. And they were not naive T cells or central memory T cells. So this just kind of demonstrated that unexposed people who do have a response are responding via effector memory cells. The authors also went a little bit further, and they examined the cross-reactivity between the SARS-CoV-2 epitope recognition and the seasonal coronavirus recognition. And they were finding that 24% of the T cell lines, which were reacting to some of these epitopes in the regular seasonal coronaviruses, were also reacting to epitopes displayed by the SARS-CoV-2 virus or similar ones. And so the amount of reactivity which was being generated was similar. And in fact, in a few people, their T cells, who weren't exposed, their T cells were actually more reactive on average than the people who had previously been infected. And so what the authors concluded was that there does appear to be a range of pre-existing memory CD4 plus T cells that are cross-reactive with comparable affinity to SARS-CoV-2 and common cold coronaviruses that regularly circulate through the human population. And they also found that a smaller percentage of them also had CD8 memory T cell response, though they didn't elaborate too much on that. They were finding that this cross-reactivity seemed to be pretty robust, and the immune response which was being generated was similar between the seasonal coronaviruses and SARS-CoV-2. And then the authors also concluded that more research is needed to see if these memory T cells play a role in the severity of disease. The limitations of this study also had relatively low power in terms of the number of donors studied. It also did not attempt to measure the effects of having these pre-existing T cells on illness severity, which would be kind of hard to do, particularly hard to do ethically. And then it really, it only really looks at, T cells and T cell memory, and it doesn't examine, the role of any other parts of the immune system in, in its potential for limiting or containing SARS-CoV-2. So are there any questions?
Yeah, I'd like to make a comment here. First of all, thank you so much. These two articles caught my attention when they came out, and I think these folks are on the right track for a deeper understanding, not only of SARS-CoV-2, but of our immune systems in general. And there's no doubt in my mind this is going to contribute to getting us out of this mess. That's number one. Number two, that you chose to present these two speaks volumes about you. You're to be commended for this is a heavy scientific lift, and you've done an excellent job. One point of note for you, the word epitope is how it's pronounced. Epitopy is, just so you know, moving forward. But again, hats off to you, my friend. That was great. Dr. Johnson, you have a comment or question?
Jessica, I'd echo everything Dr. Steiner just said. Plus, my question to you, since your depth of knowledge is really, really impressive, from review of the second article particularly, what surprises you the most as you've scoured over this article? What was it that stood out to say this cross-reactivity is surprising to me, or did it?
Well, I guess that, before all of this, I tended to have a view that was probably more like, like the standard, like media outsider view where it's like, oh, you need antibodies to protect you. And, maybe the antibodies can cross-react, but I don't really know about T cells. And so definitely when I encountered these articles, I found them to be very interesting. And I do, feel that it would also explain a lot, or it would make a lot of sense in terms of what we, know about the immune system within aging and the distribution of, who tends to get really sick and who tends to not get really sick in terms of, age, particularly with it, being a pretty, mild illness in most children. Though I know that, there's also that argument that, the cross-reactivity plays a role in kids as well, since kids are catching colds and viruses all the time.
Thank you.
Another quick comment. In my view, this presentation emphasizes a truth, and that is that vaccine science and virology are at the interface of the human condition and medicine. And many of the advances in the biochemistry laboratory or in laboratory science in general hail from the field of virology. Everything in molecular biology can eventually be traced back to virology. Viruses are so tricky, and that this is a truth that you've brought forth by presenting these papers is much appreciated by me. Dr. Schneider, you have another comment?
Actually, I have a question. Jessica, relevantly to the second publication you presented today, are they giving you the age of that people who had a SARS infection previously? Considering that it was a beginning 2002, 5 or whatever, so it's they already have only memory cells, my understanding, by the age, right? So do you have any information about how old group was, what the range for those who was with SARS infection?
I don't remember seeing anything about the age of the donors.
Okay.
I'd just like to
But-
point out to this group that the manuscripts are linked in the announcements. If you are aware of that, you can click on them and it'll take you to the papers she presented.
All right, thank you.
Ms. Boyer.
Jess, I was wondering, I saw a paper a while back that mentioned that maybe people that had been exposed to or had received, I can't remember what it was, certain flu shot during a certain time, maybe they were showing some kind of cross-reactivity and immunity to this SARS, the COVID-19. And so I'm wondering if you think that's a possibility, sort of touching on like what Dr. Staudinger said, everything's related, vaccines, the virology, the immune system. Do you think people that potentially have been exposed to prior flus or received prior flu vaccines might have some protection of some sort to this?
I think it's, theoretically possible. I think that, the flu and the coronaviruses in terms of like their sequences and potential like epidemiologies, which the immune system could respond to, probably vary a fair amount, but I don't really know enough about virology to really say that definitively. I would think that probably the best chance at cross immunity or getting some sort of cross immunity would be from having, prior other coronavirus infections, since the other coronaviruses in the coronavirus family have a certain degree of like reserved genome and reserved antigens, which the epitopes can bind to.
Dr. Schneider.
I just would like to comment on that question what Shelley asked. So all FDA-approved vaccines used for immunization of our children and immunization adults for flu. They are designed to produce antibody-mediated response. Even we know those are viral infections because antibodies, which is neutralizing antibodies, prevent reinfection of healthy cells when virus is spread. But the point is here that immune system is very smart. And when you produce something which is a surrogate form of infection, it doesn't care about it. It's antibody response and that's it. Now, immune response is actually what is optimal is a real infection. And that's real infection. You can use attenuated viruses. That's will result in normal T-cell mediated response. All others are surrogate. But we also are throwing away, it's seemingly not related issue, that any immune response to infection also generate extremely robust T regulatory cell responses. And that is not what looked now, which is very important because you train immune system to respond to that. What they will do if we have a person with robust ability to create T regulatory responses. So they will not go that shock system when everything goes out of control, because those T regulatory cells can dump overreactions, because we have a SIRS, systemic inflammatory response, and we sell compensatory anti-inflammatory CARs. Usually young people are dying in the first phase, which is a systemic inflammatory response syndrome, where it's dominated by pro-inflammatory cytokines. But elderly people are dying by compensatory anti-inflammatory, which is also sometimes leading to system which call it immune paralysis. It just doesn't work anything. So vaccination from standpoint of immunologists, if you want to hear, I'm not advocating not to do it, but if the best way is, of course, to get that infection and you will have a normal immune response. So that which provided you with the surrogate by soluble antigen, etc., I doubt that it will result in any protection for cross-reactivity, etc. That should be, from my opinion, is active immune response. And in these both studies, they compare people with documented SARS infections and COVID-19 infections, not just, all right. So, but cross-reactivity may happen because we have other coronaviruses which people just had a kind of flu-like infection, never was immunized, and that against that. That's why you have cross-reactivity with so, but I doubt that it will be with a flu virus.
So I have a question for you, Dr. Schneider. Does that imply that the Moderna vaccine that's coming, which is nothing more than the spike protein produced via, will be ineffective in your view? And would you prefer a live attenuated SARS-CoV-2?
That's what I don't know exactly Moderna because I know about Russians who already registered vaccine, principally made on the same principle. They claim that they will provide immunity for 2 years. For 2 years.
Yeah, they don't know that yet. It's too early.
No, because they did what they did. They take a Ebola virus vaccine which they developed, and after that, all other components, but all RNA, they substituted, and after that, they did this study, right? So, and it's absolutely mirrored responses which they get with the bigger population using as Ebola virus vaccine. And that's as they project. Of course, we're in critical situation and time when they project. So yes, my feeling is that it will be longer than just in protection when you inject, but probably about a couple of years, but not lifelong. Right. So if you have vaccination with the first Salks vaccine against polio, right? So that is what will be lifelong immunity. But you need to, if you take a toxoid vaccine, you need to repeat it every 10 years. So duration depends on whether it was active infection or it was a kind of surrogate reaction to something which partially mimics infection. So I know that some Koreans, South Koreans, they're doing vaccine on attenuated COVID virus. That's probably will give you long lasting if they achieve it, but nobody knows.
I would like to make one comment that I think is just confusing with this, because I agree with everything that you're saying, Dr. Schneider, that a live virus, live infection is going to give you that lifelong immunity, at least a better chance of it. But what we've seen are cases of people that have gotten infected and then gotten reinfected with COVID-19. And so that's where there's this sort of fuzziness of how is somebody able to get reinfected if they already were infected before?
Shelley, I ask just very shortly. That falls for me for category of anecdotal evidence. I did not see any publication about it.
Yeah, so, I don't accept that as fact. I just don't.
Yes, that's it. I'm glad that, Jeff, you share with me this stand, because
I appreciate it. We'll say fake news.
No, I'm not taking fake news. I don't blame somebody, but I will say, in order to prove, show me different data. It's saying that somebody was reinfected. So who knows? Because all of these reactions, what they test for existing infections, they can give you false positive as well as a false negative.
Apparently, in this country, we're having trouble even detecting the virus. Yes. For whatever reason.
I agree. I think it's these stories, though, that confuse people and that need to hear the science that backs it up, that you need to follow through with the news stories that you're hearing and make sure they're scientific facts, because the community thinks, I can get reinfected. And that's, what's driving, I think, further fear.
Shelley, I'm afraid I have a feeling that some mass media sources are making it deliberately.
Agree.
to make this case so heavy. All right,
Okay.
so yeah, but the only answer show the data, and it's immediately it stops short at this time. Thank you.
Okay, let's let Dr. Agbas chime in.
Well, I'm just probably providing you my 2 cents to Shelley's question. For those who are infected the COVID and survived, and then assuming that they build up the immunity and then they're infected, most likely their immune response was too weak, because there is a crowd infection theory, application actually, that let the people, the crowd of people will get infected, so they will build up the immunity. But everybody is different. Some people has a strong response and then develop and then get the immunity. The other one, past the virus, the infection, the survive, but they still have a weak immunity for the next wave, so they can get it. What do you think, Dr. Schneider? Am I thinking correctly?
Baki, we discussed it, I believe, 2 months ago, when we.
Yeah, I remember. Yeah.
Yeah. So when there
but
was
they don't know our discussion. Yes.
No, we discussed that how people could be reinfected. I tell you, so when immune system respond, antiviral response, they kill infected cells, lung cells. And what they do, they just stimulate apoptosis in those infected cells. And after that, cell disintegrates into apoptotic bodies. They may build up somewhere there. It's called remnants. And after that, after 3 or 4 days, person was cleared all infection and so ready to, it's again positive. Because, polymerase chain reaction, it doesn't mean what is there. You just get it, lyse it, and boom, you got. So it's technically, there are no infected cells. There are these remnants, which is apoptotic bodies, which are waiting for to be scavenged by macrophages, clear it and substitute it. Right, so if you have a significant level of damage, that will take some time. And therefore, I think that is a case for reinfection where some apoptotic bodies appeared, so in the sample, and they give you positive result. That is what might be scenario.
Okay. I see we're at the top of the hour, and obviously, student Dr. Jackson, you've generated a robust discussion. Congratulations again on choosing some hot papers. Published last week. My goodness. Again, a yeoman's job. Let's unmask and thank our speaker.
Thank you, Jess. You're also in a full rotation right now, so we appreciate you participating and putting in this effort. Amazing job.
Take care, people, and look forward to future coming events on the MKRC Canvas page, and rest assured we'll announce it. Take care, people. Have a great day.
Thank you.
Take care.
Jess.
Yes.
I sent you an email. I was hoping you could share your slides. They were so good. I'd like to put them on your MKRC Canvas page. Okay?
Sure. I can do that. It'll probably be this afternoon because I have to rush back to my rotation, but.
No worries.
Okay.
Either through Google Docs or just email me the PowerPoint.
Okay.
Thank you so much.
I can do that.
Bye-bye.
All right. Thank you.